Hello, thank you. I appreciate this opportunity to represent the USDA and the ARS and some of the sort of sustainable approaches we have to cotton control and specifically boll weevil eradication efforts. So my name is Zachary Cohen. I'm a postdoc here at the Southern Plains Research Center in the cotton control unit. And my talk is actually going to be entitled Differential Gene Expression, Weevils, Gene Targets for Sustainable Boll Weevil Control. So there's Elmer Fudd. I have my USDA email and my GitHub username there in case you want to follow up. Any questions? Okay, so a little bit of background about this insect. It's called the boll weevil because it actually develops the larvae and pupae within the cotton boll. But it wasn't a major issue for cotton production until the early 20th century. We believe that it expanded from Mexico throughout the southern Texas sort of tip there in the border, all the way eastward in a very short amount of time. And it was very impactful for cotton production. And it sort of motivated a contemporary eradication procedure, which is a zero tolerance threshold. So if they find one weevil in the field, they treat the whole field with organophosphates, which we know are, generalist insecticides that are pretty nasty for all animals with a nervous system. Now, this is not to be confused with the thurberiae weevil, which is a subspecies relative to grandis that actually has a preference, presumably for the thurberiae plant, which is another gossypium closely related to upland cotton and the cultivated cotton, but is not agriculturally relevant. These two plants are sympatric here. They live in the same region of the southwest and Mexico. Obviously, this sort of Etsy looking project in the upper right here, this is cotton actually as it is in the field before harvest, sort of in the rows. And then this is the thurberiae plant here. Those are flowers, not cotton balls. So these weevils are subspecies, and the boll weevil prefers cotton, and the thurberiae weevil is found on thurberiae. But there's actually a difference in the geography here, more so than the host preference. So I say that this insect is reviled and revered. Obviously, it's reviled by cotton farmers, given the impact it had on production and yield. Even to this day, it's very costly. As soon as a weevil's found, the eradication effort is expensive, but it's also revered because as cotton sort of crop crashed, alternative plants and produce were grown and cultivated in the South. So we've got, peanuts and sweet potato and yams and other sort of ubiquitous foods of the South became more prevalent and more easily grown. So we see the statue here from a town in Georgia showing that, and here's a peanut farmer. So with these whole genome sequences, what can we do with them? What is the utility of having this? Well, obviously it's an emerging system here with a high quality reference. So understanding sort of basic biology, basic evolution here, which are like the greener bubbles here, 6 and 7, is relevant. Just in understanding how diversity of weevils, which is the most specious taxon on the planet, has come about. And then more applied and like specific, more relevant to ARS and growers are, how can we better control this weevil? How can we determine the origin of the weevils that are found in the field, if they're sort of endemic to that region, or if they're sort of colonizing or spilling over from other non-cultivated sources or local areas. A little bit again of the history of the eradication. So I mentioned that it was a problem in the early 20th century, but it wasn't until the middle of the 20th that the USDA and local government established this eradication effort, which is zero tolerance. Any weevil they find, they really control intensely that field. So these are the zones here. Southern Texas is still sort of a chronic area where active eradication is occurring. And we want to, again, better identify novel targets to sort of limit the consequences of the contemporary procedures that we have for eradication. And the whole genome sequences can help us do that. So Razek et al., which is a prior whole genome effort from last year, produced a moderately good assembly. It was about 2/3 of what we expected based on Busco's and the C value of the weight of the DNA. But our assemblies are obviously much more contiguous. We had sequel2 PacBio read data, the hi-fi sequel2, as well as high-C chromatin conformation, which is what this contact map here, this dot plot on the right represents, producing a very, very highly contiguous assembly. Using some SNP data from Razek et al., we were able to they were able to determine that the weevils sort of clustered in these two geographically distinct regions that were not necessarily associated with a host plant. So here are some of the metrics from the assembly that we presented in our genome announcement paper, hopefully coming out in G3 relatively soon. And I generated gene model predictions using breaker pipeline, mass repeats, identified repeat elements. And we see very similar distribution of repeats and total gene content between these two subspecies assemblies. And we wanted to identify, using transcriptome data, how genes were sort of changing in expression throughout the development of the Bol Weevil. So the larvae and the adult are the active, impactful stages of development for cotton. Obviously the PP are more or less inert, even though there's a lot of interesting things going on there. We wanted to sort of focus on larvae and adult. So if we look at sort of this heat map here, these are all the genes sort of throughout development, and then these are the different expression patterns. We focus on expression patterns 3 and 4 here, and these are some genes that were the most differentially expressed. And there were about 2,500 genes that fell in this category, and we grouped them by type and function, which were associated with all these other sort of coarser grain categories, which are novel targets and hopefully can help us better control the weevil in a sustainable way. So I appreciate your time and thank you for this opportunity.